thanks so much for inviting me to this meeting. It's really, really exciting to be part of this event, whatever form it's taking. And I feel very, very privileged to speak a bit about what I have been doing in the last few years about 3D genomics. So this talk is about networks, and it's actually great that Marcelo before me gave a really nice introduction. So I'm just not going to bother too much with the introduction, I guess. But basically, just to remind you that there are structures at many different scales. And let's say that in the first talk, Marcelo spoke so much about relatively small scale structures, and I will try to give an overview that spans more length scales in a lot less detail. So like he was saying before, sorry, let me just right. So the most apparent structures when you look at chromatin are obviously TADs. We all have heard about TADs. And I just wanted to say that the great opportunities of looking at single cell is also helping us really understand how the formation and the function of these TADs can be associated either with an ensemble phenomenon or with single cells. And also I wanted to bring your attention to the fact that single cell Hi-C has been performed across the cell cycle, and people were able to identify specific patterns depending on the phase of the cell cycle they were looking at. But really, this talk is about an approach which is based on chromatin networks that I have been using to study DNA structure in different scenarios. So the main idea, which is not completely novel, I agree, is that genomic coordinates are normally seen as a line. But actually, this is a description that we have idealized in our head about a sort of spaghetti ball or a three-dimensional organization of chromatin inside the nucleus. And so to me, maybe it's just a bias. This looks very much like a network. So using networks to represent chromatin has become very common. So we see here an overview of methods from literally 10 years ago to the most recent, and I'm sure there's many more. This is just a summary. But basically, I'm going to try to convince you that it's a quite powerful way to actually describe what we have in these different types of data sets, whether they're from microscopy or more typically from Hi-C. So what I did a few years ago was actually develop a method to look at chromatin associativity of epigenetic marks. So we started by having a large set of ChIP-seq on mouse embryonic stem cells. And we collaborated with Peter Fraser, who had just produced a chromatin interaction network, also in mouse embryonic stem cells, using something called promoter capture Hi-C. And I will go back to describing exactly what this is. Basically, the idea is that if you represent the chromatin structure as a network, and the contacts, rather than looking like arcs in a linear genome, becomes basically edges in this three-dimensional network, you can also project lots of different information, like, for example, all the ChIP-seq peaks on top of this network. And this represents a color image of the network. So once you do that, it just occurs to me that we can do something very simple, which is estimating whether these features are assortative on the network. And this basically just means whether they form clusters. So assortativity is a network property that is often used in social sciences, but applied to chromatin, it's just asking whether the regions that have a specific feature tend to interact with each other and away from the rest or not. In this case, the blue nodes interact either with blue nodes or with white nodes with an equal probability. And of course, you can also have negative assortativity, which would mean that basically a blue node always interacts with a white node. So what I have been starting to do is studying this CHAS, which is abbreviation for chromatin assortativity, in these plots that basically show us what is the assortativity we observe on a specific data set, on a specific network, and what is the random expectation for this assortativity that we expect based on randomizations that we can perform on the data. So these type of plots you will see appearing very often. But the second thing that I wanted to introduce is the fact that there is a specific version of Hi-C which we really like, which is called promoter capture Hi-C. So you all know what Hi-C is, but basically promoter capture Hi-C is just a Hi-C biased to capturing promoter interactions. So either promoter-promoter interactions or promoter interactions with their enhancers. So basically this is an example of a promoter capture Hi-C data set. And using the method Chicago that was developed some time ago by some colleagues in Abraham, you can basically identify which ones of these interactions are statistically significant. And once you have done that, you can reconstruct this P-chick network that can be decomposed into a PP subnetwork. So these are interactions between promoters, and a promoter other end subnetwork that is here represented in blue. So you can either decide to look at the whole thing together or to look at the two subnetworks separately. And actually, this is what was the basis of my first paper on this topic, where what we decided to do was take all these different ChIP-seq data sets we had been collecting from muscle-embryonic stem cells and look at the assortativity of each one of these transcription factors and chromatin marks, either in the promoter other end networks or on the promoter-promoter network. And we started observing interesting things. I'm going to go through this quite quickly because it's published, but just to give you an idea, we were able to see that polycomb features were actually highly assortative, and especially they were more or less on the diagonal of these plots, which means that polycomb is involved in either, well, in both promoter other end contacts and promoter-promoter contacts. This was not really the case for other features that were actually polymerase II features. And here we have 5 different features that refer to polymerase II because they represent different states of the polymerase II as it proceeds in its transcriptional first initiation and then elongation. So what we can see for these polymerase II features is that all of them are assortative in the promoter-promoter network, meaning that basically polymerase II is forming clusters in the subnetwork of promoters. So promoters that have pol2 are interacting more strongly with each other. But at the same time, we see a great variability in the assortativity in the promoter other end contact. And this means that only the polymerase form that is elongating, which is this one up here, RNAP2S2P, is the one that is actually, I couldn't say bridging, but it seems to be mediating the contact between promoters and other ends. I don't like to talk about bridging and mediating because I really don't know if this is a causal relationship, obviously. But basically, it seems that the RNA polymerase, which is elongating, is present when the promoters are interacting with other ends, whereas it's making clusters in this type of interactions. On the contrary, we can also see some things that have negative assortativity. For example, we were very happy to see that this H3K4-ME3, which is a mark of active promoters, was actually negatively assortative. And this is actually a sign that we are on the right track, because this means that in a promoter-promoter interaction network, there is assortativity, and we know that actually active promoters tend to interact with each other. Whereas in the promoter other end interactions, because one of the two sides of this interaction is another end that will not have this mark, we see negative assortativity. So if you want, this is obvious, but it helped us understand that actually using these plots and this metric, we could actually study what are the principles that might be behind chromatin organization. So we were able to reproduce these things that were known, and actually polycomb was just at the time being recognized as a main regulator of 3D chromatin structure, because Peter Fraser the same summer basically produced a paper where they knocked out polycomb and realized that the network would fall to pieces effectively. So this was the beginning of the paper, and then the second part of the paper was actually presenting an analysis of network properties of the chromatin network by separating the nodes that were either occupied by polycomb or by polymerase II features. So we did a sort of community detection analysis of the network. We defined some things like betweenness centrality, bridgeness, and basically the main findings was that polycomb is a stable hub, which basically means that it has very high interaction, very high number of degrees, so very high number of contacts with other elements, and stable in the sense that according to the bridgeness and betweenness centrality that we were able to measure, we could see that it was probably making interactions that were fixed with the same element. Now, of course, this becomes a bit philosophical, stable across what, because this was an ensemble data set. It was a population promoter capture high C. So we're not able to say exactly what this means, but this could mean across cells. It could mean in time, because we are not really sure what this means, but it's different from what we saw for RNAP2. So polymerase II is also a hub. It interacts with many genes, or with many, many nodes interact together. But they seem to be less stable interactions. So this could represent the fact that the same promoter might interact with different either promoters or also with different enhancers. Like what Marcelo was showing is that clearly there are a lot of interactions between promoters and enhancers that can be established and dynamic across either cells or time. And the interesting thing was, of course, that this other form of polymerase II, which is related to the elongation process of transcription, was actually not a hub at all. And it remained very peripheral as estimated by our network measures. So what we were able to do was actually predict that this S2P form would be peripheral. And I was very happy to see that experimentally, some people were able to observe that actually CDK9, which is the kinase that phosphorylates this form of polymerase II, remains at a sort of periphery of what could be considered a transcriptional factory. So if we have different promoters that come together in a transcriptional factory, they seem to be leaving the factory in the moment when elongation is taking place. So this is all slightly hand-wavy, but I think that by looking at these networks and the network properties, we were able to make some observations about the important role of the elongating form of polymerase II that seem to be confirmed by experimental observations. So just to continue, when I started studying this, we were doing it with my own scripts and following just my ideas. But we decided to do it a bit more systematically, and so we started developing some tools. So the first tool that I'd like to present is GardenNet, which is actually a web browser, effectively. So if you go to this website, you can basically see the chromatin structure of different cell types represented as a network. And this is entirely interactive so that you can click on links, you can click on nodes. And actually here you have a zoomed out version of this part of the network. And you can calculate chromatin assortativity for a lot of features that are already loaded. But I'd like to point out that you can also interactively use this system to upload your own data and calculate assortativity of your own features. So as you see, there are quite a few data sets already loaded, but you're very welcome to try it and actually contact me if you would like to try or you have any issues. So we thought that we wanted to at least give the community an opportunity to try and represent these chromatin structures using this network system. And the second part of this tool section of the talk will describe Chaser, which is actually an R package, which is behind the calculations of chromatin assortativity. So basically, Chaser takes a 3D network as an input. And I should probably mention after the talk from Marcelo that this input doesn't have to be a high-C network. It can be a high-M network. It can be any network based on chromatin that you have identified. But the problem is that you have to have chromatin coordinates, so it's not really any network. But once you have chromatin coordinates for your interacting fragments, you can combine them with chromatin features defined on the same genome, of course. And then the package Chaser will basically map the features on the network and calculate assortativity with all the different randomizations. And for those who have seen the first version of assortativity analysis, we have really greatly improved the efficiency of the network storage and processing. And this was a great contribution from Emanuele Raineri from Senag in Barcelona. We have also improved our methods for randomizing these networks to estimate really the importance of the 3D component of the structure in determining the assortativity of specific features. And we were also able to extend the concept of assortativity to pairs of features. So either we might want to see the assortativity of two features being on the same fragment, or we want to see the correlation of features that are occurring on the two opposite sides of the edge. So if you're interested in this, please make sure to see the paper and you can find the code here. But really what I would like to spend some time on is talking about what we have done with these tools. So I will discuss a little bit about replication because we spoke about transcription, and I guess this is what most people are concentrating on, the big question of is the functional role of chromatin structure to regulate transcription, or is it the other way around? But I'd like to talk about replication. So replication in 3D, I mean, I'm really not an expert, so I will give you an overview, but a sort of ignorant overview. It's actually a very interesting process that involves the stochastic firing of lots of origins across the genome. So there are debates about how stochastic it is and the details. but basically different origins at different places in the genome will start replicating at different, during one replication. And basically, it has been observed for a long time that these replication origins come together in what are called replication clusters. And basically, the origins that are in the replication clusters would activate, and then replication would spread away from them to completely replicate the genome properly. Because this is a very important thing. If we have mistakes in this replication, obviously this has huge consequences in chromosomal aberrations and aneuploidy, and horrible things can happen. But basically, the idea is that cohesin, which is omnipresent and doing lots of different things, would mediate the replicon loops that basically keep together these replication domains. And it has also been identified by Gilbert and co-workers that these replication domains coincide quite nicely with TADs. So the idea we have is that basically 3D structure is also intimately related to replication, and that basically you would have the parts of chromatin that are more active that would be early replicated. Early just means that basically at the beginning of a replication cycle, that's where replication starts. And these regions are also more dense in replication origins, whereas the regions that are late replicating would be having less origins that fire with less efficiency. And these will be regions that contain less genes or less important genes. So what we have done in collaboration with Juan Mendez at CNIO in Madrid, when I was in Madrid a long time ago, was actually produce a new origin map in mouse embryonic stem cells. So we used short nascent strand sequencing, which is a method to basically sequence the little pieces of DNA that are being replicated. And interestingly, we were able to compare a wild type mouse embryonic stem cell together with cells that were treated by afidicolin, so APH is for acronym, which is basically a treatment that slows down the replication forks and generates mild replication stress. But basically the effect of this condition was to hyperactivate the origins, because the cell reacts to the stress by trying to rescue its replication program. And so extra origins are activated. And the third condition was actually an overexpression of CDC6, which is a very important licensing factor, which also increases the number and activity of replication origins. So now I'd like to define the concept of origin efficiency, which is actually not so complicated, but it refers to the number of reads that we see in our short nascent strand sequencing assay, but it also represents basically how many origins are being used in a population during one replication cycle. And it's very important to mention that this efficiency can be estimated even from bulk data, because it's not like transcription. You can have the same cell transcribing the same genes many times, but you cannot have the DNA being replicated many times in the same cell. That would create enormous problems. So basically the idea is that we have a catalog of replication origins in the different conditions, and we were able to define what we call responsive origins, that are the ones that are activated only in APH or only on CDC6 conditions, or in both these conditions, but were not present in the wild type. So some of the findings were the fact that obviously APH and CDC6 activated more origins. And also we saw that many of the origins that were suddenly activated in APH and CDC6 actually have a small peak in wild type. This is due to the fact that, this being a sequencing experiment, we are kind of tied to the fact of performing peak detection to be able to define origins. And so sometimes we see that if in a responsive condition, if in a stress condition, an origin is responsive, it's because this tiny peak in the wild type is not actually defined a peak. But we think that actually some of these origins are also very, very mildly activated in the wild type. So the question was, can we now relate all of this to 3D structure in concrete terms, meaning putting it as a projection on top of the promoter-promoter interaction network we have from mouse embryonic stem cells? So now I will focus on the promoter-promoter interaction network purely because origins are enriched in promoters, but I will come back to this later and show you that this is not limited to the promoter-promoter network. So here you have an idea of how we basically map the origin presence on top of fragments to be able to map origins on top of the network. And what we noticed is that actually fragments containing origins can interact both intra but also intertads. And indeed, there's peaks of very long range interactions around 10 to the 7 or 10 to the 8 bases. And these are basically promoters that interact with each other and contain origins. So we think that actually these replication factories that were described in a small genomic scale are probably coming together in larger structures. So another finding that we presented in this paper was the fact that origins in nodes with high degree have earlier replication timing and higher efficiency. So what you can see here is basically a tiny subset of this replication origin network. And you can see that the nodes that have more interactions tend to be with higher efficiency. And you can see here a correlation between the efficiency and the degree. And similar things when you look at the replication timing, because of course the replication timing is perfectly correlated with efficiency and also with degree, because the regions that are more connected tend to be open chromatin, which is traditionally associated with early replication timing. So we see all of these correlations. But then we wanted to go back to what are the patterns of efficiency in this network? Because it is one thing to say that there's a correlation in origin efficiency, but is it actually a statistically significant correlation? So what we looked at in two different data sets was basically the assortativity of efficiency of origin across our different data sets. So constitutive is basically the origins that are seen in all data sets. And you can see here, the red points are the measured assortativities in the data sets, and these empty black circles are the randomizations. You can see that all of our data sets have assortativity values that are higher than what you would expect by a distance preserving randomization. So this suggests that the 3D contacts across the different genomic regions are important to determine the assortativity of these origins, and not just the fact that there might be linear domains that have similar origin efficiency. So we were looking for other data sets because actually I have mentioned to you that promoter capture high C is inherently biased to promoters being very central because it's a promoter-centered network. But we had the opportunity of finding a different network that would also give us a promoter-biased view of the chromatin contacts. And so we took the data from Bonev et al. In 2017, which is a very, very high-depth sequencing of high-C experiment in mouse embryonic stem cell. And we were able, with collaboration from Giorgio in the Cavalli's group, we were able to perform a sort of virtual promoter capture high-C. This means that we take the very high resolution, the deep high-C data, and we basically look only at interactions that involve promoter elements. So when we do that, we are actually able to reconstruct similar analysis, and we still see assortativity of our data set for origin efficiency compared to random. So we really think that if we take a promoter-centered version of a chromatin network, efficiency of origins is assortative. But we were still not too happy because both of these approaches were promoter-centered, and even though origins are enriched in promoters, there are also origins away from promoters. So the question was, can we now look at all origins? And so we took again this very deep high-C data set from Bonev et al., and we produced a virtual origin capture high-C. So we looked at interactions that involved only our little origin fragments. So we defined the genomic regions that had origins and expanded a little bit around, and actually generated this network where each node is an origin or a little fragment containing an origin. And again, we can see the assortativity. Now, I didn't put the number, but you can see the assortativity because the blue nodes that are the origins that have higher efficiency and tend to connect to other blue nodes or green nodes, whereas the yellow nodes are actually in other regions of the network. Now, interestingly, since origins are enriched in promoters, obviously a lot of origins in this network are actually promoters, but we actually did the exercise of excluding the promoter nodes, and we still see assortativity of origin efficiency. So the conclusion so far is that assortativity of origin efficiency cannot be explained only by proximity with promoters, even though, of course, promoters have a really big importance in defining, how active origins are, or at least again, I don't want to say causality, so I don't know which way around it is, but certainly there is an association between origins and promoters. But beyond this association, the network structure can actually determine contacts between origins that have similar efficiency. So what this brought us to conclude was that basically origin efficiency is something that could be looked at in a global manner. So we could look at single fragments that could represent single origins, but we even averaged efficiency of origins across entire TADs, and we were able to show that even origin efficiency at the TAD level is assortative on a TAD network. So this really suggests that there is a small-scale organization of origins into replication factories, but that these factories would actually come into contact together across TADs to produce much larger replication structures. And actually, this brought us to some other findings we had in our GardenNet paper, where we saw that, of course, replication timing is extremely assortative. This was not really a surprise because, as I said, it's been years that it's known that replication is really highly associated to TAD structure. But we were able to show that if you don't even consider TADs and just look at the assortativity of the RT value, it is very, very high. So there are literally domains of early replication and late replication. And this brought us the question of, okay, we have started looking at these 3D structures because we call them transcriptional factories. At least promoter capture Hi-C was devised to be able to identify and study transcriptional factories. But now we see that replication features have a higher assortativity and a very, very global organization of replication seems to be in place. So the question for me is still completely open. But given the numbers, I'm starting to think that it's more a replication structure and a replication function. It could be that the transcription is a sort of consequence. But of course, this brings up all the problems that replication is not related to the phenotypes during the life of the cell. So I want to be provocative here. Okay, now another question that started bothering us was that we had, as I said before in our replication study, we had used mild replicative stress to basically reactivate origins that might have been dormant in the wild type. But we recently performed an experiment with Lila Pourtot, who just defended her PhD thesis in the lab of Valérie Bergoglio and here at the CRCT in Toulouse. So they performed an experiment where they used mild APH treatment to provoke replication timing changes across some cell lines. And they were definitely able to see some regions being delayed and even some regions being advanced in their replication timing, which was quite surprising. So these changes in replication timing can span very different sized regions. Normally they're quite big regions, especially for delays and in fact, there are different sizes for delays and advances. But basically, the interesting part is that even though they were finding it very hard to see the effect of these changes in replication timing on the same cells that were being measured, they found that some of these changes in replication timing could be inherited by the next generation of cells that come from the cell that was originally stressed with APH. So now this opens a lot of interesting questions because we would have to know if the mild replicative stress that they applied could change 3D structure, because a replication timing change is very likely associated with a change in chromatin structure. The reason that we think this might be happening is first, that these advanced regions are enriched in HUK9-ME3 and in lamin B. And these regions, so the treatment was shown to increase the accessibility of chromatin through ATAC-seq. So now we wanted to see if the changes are accompanied by 3D chromatin structure rearrangements. And this is very preliminary, but we saw that the replication timing difference across the network is actually assortative in a promoter capture Hi-C network. This is specifically for the HCT116. So you can see here that there are regions that are advanced that are actually in 3D contact with each other. Now, it's true that these regions are really big, so we haven't been able to find interchromosomal interactions, for example, and it could be that this is very much due to the linear extension of these regions. So obviously this is still a polymer, so things that are close to each other linearly would interact with each other more. But we think, given our estimates of assortativity and randomizations that take into account the distance, we think that there is assortativity of the difference in replication timing. So this is not entirely surprising, because the lab of Gaël Legube has actually shown that DNA damage can actually, inducing DNA double-strand breaks and very serious DNA damage can produce a rearrangement of the 3D structure. So the question here is it even possible that with this, what we think mild acetylcholine stress, we are actually removing some regions from the laminal compartment and moving it towards an open compartment? This is still a very much open question, but it's very interesting, and I think it will require further work to be addressed. So I just wanted to conclude going a little bit beyond replication. I've shown that basically replication timing is very assortative. You can see here these blobs that form. And also efficiency of origins of replication is assortative. So again, you have little clusters across the genome. But then the question is there something more important about this that could affect the variability of gene expression in these regions? And actually, there's a very interesting paper from some of my old colleagues in CNIO about relating the chromatin states, replication timing, gene density, the age of genes, and phenotypes. So to address all of this, I think will take some time, but I'm hoping to do this in the future and try to understand how the replication structure of the genome could actually impact phenotypes. So just to conclude, one part of my work that I haven't been mentioning at all is actually related to studying hematopoietic variability. So some years ago, we were involved in this blueprint project where we produced a lot of epigenomes of reference for different hematopoietic cell types, so blood cell types. And we were lucky enough to have these 17 chromatin contact networks from different hematopoietic primary cells. So we have done some work on studying the variability of gene expression and methylation across many different individuals as part of this project. So this was 125 individuals for which gene expression, DNA methylation, and also all of the genetics was performed. And we were able to estimate expression variability with a robust measure that actually removes the dependence on the mean. So we also propose that some of the determinants of this variability could be related not only to the local chromatin state, which I think has already been discussed, not only to the position in a potential gene regulatory network, which I think has been mentioned for years now, but there could also be an impact of the 3D chromatin contact, not only the local one, but literally the global organization of chromatin to impact variability. And so we started looking at this with Flavien Renal in my lab. This is completely unpublished and very raw. But we are able to identify different classes of gene expression variability. So here you see 10 different bins of EV, which is expression variability. And we see that basically polycomb target genes are enriched in the bins that have high expression variability. And actually, we see an almost inverted correlation with genes associated to super enhancers. And obviously, we were looking at chromatin assortativity of the expression variability, and we see that expression variability is also assortative in the 3 types of cells that we looked at here. Even more so, the genes that are associated to super enhancers are assortative in the 3 cell types. And so basically, we think that polycomb and super enhancers are basically 2 opposite forces that can shape the amount of variability in gene expression. But both of these categories of genes, so either the very variable or the very stable, are assortative. And this could basically create a global organization of the 3D genome. Just to mention something else that is unpublished, we were able to look at assortativity of expression variability even in single cells. This was done in mouse embryonic stem cells, but we're now hoping to reproduce it in the immune cells. And we also see associativity of single cell variability. So we think that really variability at many different levels is or could be determined by a 3D global pattern across the chromatin. So to mention a few perspectives to conclude, I think that chromatin heterogeneity, plasticity, and stemness, they're all very big words, they might all be connected and everything revolves around chromatin. So this slide used to be original some years ago, but now there are plenty of papers that are coming out to basically strengthen this type of statements. And what I'm interested in is basically trying to understand how we can use chromatin networks and their structures to understand what is the process that creates heterogeneity or reduces it during the process of differentiation. And this is intimately connected to plasticity, which I interpret as basically the capacity of cells to adapt their phenotype to their environment. So I can make some sort of hypothesis that the pluripotent cells we start with basically would contain a high level of isogenic heterogeneity, so phenotypic heterogeneity, and they are subject to large fluctuations in their transcription, which could represent a sort of motion across this landscape that is more free up here. And this definitely represents plasticity, so that these cells can basically decide how to respond to different signals. But as the cells differentiate and we go down this landscape, we will have reduced heterogeneity and plasticity and achieve a well-defined phenotype. So this reminded me of some work that I did years ago about the effect of stress response on protein-protein interaction networks. And we saw that the network structure was actually reflecting the changes in the expression profiles that were created by the stress response. So just to really conclude about what I'm doing this for, because now I'm in a cancer research institute, we are studying variability across patients. And the main question we have is to understand why certain patients respond to some therapies that basically act to reestablish some sort of equilibrium that would impede cancer growth inside a tumor. And we would like to see if we can understand the formation of these cells that are called tumor-associated macrophages that end up protecting cancer cells against our body's defenses and also against the therapies. So basically, I have said that in Blueprint, we were able to investigate the variability in healthy people in many different levels. And now the quest for the next few years will actually be to study what happens in patients when they become patients to affect this variability, and actually study how variability in the immune cells of these patients could potentially affect the formation of these cells that are creating resistance to therapies and definitely promoting tumor development in these patients. So obviously, the way we plan to do it is to look at the 3D network structure of these cells, and we have some preliminary data that genes that are differentially expressed between these different types of cells that actually produce the resistance in the cancer are assortative in the respective chromatin structures for the cell type. So with this, I've certainly run out of time, but I wanted to acknowledge all the help I got from my team. So this is my team, especially Flavien Reinal and Miguel Madrid-Mancia, who are responsible mostly for the chromatin work that's being carried out in my team. And some collaborators from Barcelona Supercomputing Center, where I also am co-affiliated. Senag, of course, because Emanuele was really key for the chromatin assortativity paper. Some old collaborators who were at the beginnings of chromatin assortativity. Simone Ecker, who is my main collaborator for gene expression and phenotypic variability. And then collaborators from Bebraham, Senio, and other places in Madrid for the replication study. And obviously, thanks to all my funding. And thank you for your attention.
Thank you, Vera. Thank you so much for your presentation. So we have questions for, we have time for a few questions. So I will start. I like this idea of, I mean, basically when you do have this kind of memory that pass through the 3D organization, I was wondering if you have experiments to test that hypothesis, and in particular, did you, can you do perturbation in your systems regarding key replication proteins or complexes that can affect the 3D organization? And also, are you sure that is not changing in the chromatin landscape, basically in the epigenetic, that is not affecting the 3D? That's really the memory is in the 3D, or it's passed to 3D via epigenetics?
So that is a very, very complicated question to answer. I think sticking to the fact of do we have experiments, for the moment, no, but it would be certainly interesting. And, you know, this work on trying to produce replication timing changes was quite surprising to me, because I didn't think that it would be so easy to change the replication timing program. Of course, we should basically perform, like you say, traditional epigenetic analysis, so the chromatin marks, the standards, to see what happens. But I think that you might be right that the memory might be in the marks, because I think that the 3D organization, if you think about the fact that you can replicate it in the next generation, there has to be a clue as to how to reproduce the 3D structure. I think it's probably a combination of 3D marks, but also maybe the sequence itself. You know, a lot of people are going back to the idea that the sequence would be determining both for the epigenomic modification and for the 3D structures. And also there are some papers about sequence motifs that are associated both to origins of replication and also to replication timing programs. So I think that it's an interesting question, and unfortunately, experimentally, my lab does other things for the moment, but we have collaborators, so I'll try to assist them into helping.
Okay, thank you. So we have some questions. So one from Marco Di Stefano. So this question is about the input. So why do you need to filter for promoter-promoter interactions? And do you use any normalization process to induce the PP network, for example?
Okay, so why I don't filter for PP interactions. In the promoter capture IC network, it's the technique itself that filters for interactions involving promoters, so either promoter-promoter or promoter other end. When I look at the Cavalli dataset to do the in silico promoter capture IC, I wanted to reproduce what I had in the promoter capture IC just to be able to compare the two. So that was obviously a very deliberate filtering, if you want. Now, when we looked at the Cavalli data, we also did origin capture IC. So there we did not bias to promoter-promoter. Maybe the reason you're asking me this is why don't I use high C as it is? So the reason is that if you use high C data as is or looking for loops, you need to have very, very deep high C to be able to see long range interactions. And whenever you have methods that do not filter away a lot of interactions, you always get completely swamped by short scale interactions. I have used ChiaPet datasets, which are actually very nice because you focus on interactions that are mediated only by a protein, but that means you don't have the rest of the genome to bother you. And I've even tried using DNA's hypersensitivity capture high C, which is supposed to capture the open regions of chromatin and their contacts. But even that captures too much. And so again, if you don't sequence deep enough, you will completely lose the long-range interactions. Now, I don't really want to raise issues, but there are promoter capture high C datasets published for 27 different cell types. And the people who produced the data basically use statistics to eliminate all the interactions that were above 2 megabases of range. That to me is what in English you say throwing the baby out with the water of the bath, right? So it's a choice, right? But if you're interested in large scale and global organization, throwing away those interactions is really not a good idea. So you have to kind of go for the techniques that allow you to preserve the long range interaction. That's why. About the normalization. Yeah. Actually, what I call randomization is basically normalization. If you do the Z-score of your real value and the randomization, that's your normalized associativity.
Okay, so we have a question from Pierre Caron. Are the responsive origin in response to APH enriched for specific features, such as topoisomerase or G4 structure?
Okay, so I haven't done the enrichment for G4 loops and topoisomerases. I can try that, but yeah, we haven't done it.
Okay, another question regarding this APH. from a question from Spark. Why APH-induced blocking of polymerase is increasing the number of origin? Can it be due to an increase in activation of gene networks involved in stress?
Well, okay, I don't really know if it's related to the activation of the stress, as in genes that are basically promoting the response to the stress. I think the idea is that because of this replication stress and the fork stalling, the other origins are rescuing the replication process, so that basically the dormant origins are activated to make up for the fact that the main origins are stalled. But I'm not an expert in replication, so I cannot answer that very, very satisfying.
Thank you. Marcelo, do you want to ask your question?
Sure, thanks. Very nice talk, Vera. So, so I had a question regarding the data sets that are now available for time-resolved high-C from Blobel or even for Peter himself. Have you tried to do this network during, for example, different stages of interface?
Not yet.
Okay.
I haven't tried. I'd love to, but the problem is I have a separate life studying tumor microenvironments and interactions of cells in tissues, but I'd love to try. So we have plenty of ideas to apply this, and I really would love to. Yeah, it's a good idea.
Just a last question from Kuljit Sandhu. Is there any rich club in any of the chromatin networks? And if so, what can be the significance?
Right. So I haven't really looked at rich clubs in specific. Actually, Kuljit is one of the first people to look at network structures in Chromatin Networks the way that we have done. I haven't looked specifically at that. There's so much to look at. So I'm sure that there are communities, and recently there's a paper on TAF cliques, which I think is very relevant and very much related to what we have seen in replication. I just think that there are different approaches that are basically achieving the same results in different ways. But it's kind of reassuring, right? So it's confirmation in science, it's good.